chapter 4 on the anatomy of cells. So there's two main categories of cells. We have prokaryotic cells and eukaryotic cells. Now all cells have DNA, which carries their genetic information. All cells have cell membrane, which has cytosol inside of it. And all cells have ribosomes, which help make the proteins. The type of ribosome is different in prokaryotic and eukaryotic cells, but they both have ribosomes. Now, prokaryotes, these are all your bacterial cells. They're smaller than eukaryotes. They only have one circular chromosome. So all of their DNA is on one chromosome and it's circular. And that DNA is not membrane bound. So that means that there's no nucleus, there's no nuclear membrane. The DNA in prokaryotes is not wrapped around histones. There's no membrane-bound organelles. So if you look over here, you don't see like any organelles, right? You don't see mitochondria, etc. You just see the ribosomes. That's all these little red dots are the ribosomes. You see your DNA here, all inside the cytoplasm. Prokaryotes have cell walls, and those cell walls are made of peptidoglycan. And they reproduce through binary fission. On the other hand, we have eukaryotes. Eukaryotes are much larger. They have multiple linear chromosomes. So instead of being circular, now they're linear. For example, the human has 46 different chromosomes. So we have multiple linear chromosomes, and those chromosomes are going to be held inside of the nuclear envelope, or the DNA will be inside of the nucleus. And the DNA is wrapped around these proteins called histones. So here's our DNA double helix you're familiar with. If we take 8 of these histone proteins, then that's going to wrap, the DNA is going to wrap around 8 of those. It's going to do 2 wraps around. Then we're going to have 8 more histones. It's going to do 2 wraps around that, and then around the next one, and so far, and etc. until we get it all bundled up together. So that helps to keep our DNA more organized. We also have lots of membrane-bound organelles in eukaryotes. So we have like the ER, endoplasmic reticulum. We have Golgi body. We see mitochondria, lots of them. Eukaryotes may or may not have cell walls. So like human cells do not have cell walls. As we talked about in chapter 1, chitin is in the cell walls of fungi. Cellulose is in the cell walls of algae. And eukaryotes reproduce by mitosis. So now focusing back on prokaryotes. So most of our discussion moving forward will be on prokaryotes. So there's 3 basic common shapes for bacteria or prokaryotes. We have bacillus shape or rod shape. That's what's shown here. It's either longer than they are round. Then we have coccus shape. So those are round bacteria. And the third most common one is spiral. There's also star-shaped and rectangular shapes. Those are less common though. And then there's also, those cells can be arranged differently. So here's lots of examples of cell shape and arrangement. So here we have our 3 most common shapes of the cell. So you're going to look at each individual cell and look at its shape. So if it's round, that's going to be a coccus. If it's a rod shape, if it's longer than it is around, then that's going to be a bacillus. And then here we have some spiral shapes. And then the way that those cells arrange themselves with other cells is what we call cell arrangement. So you can have just 2 of them. If it's coccus, that's going to be diplococci. Strepto is usually going to mean like arranged in a line. So we see the streptococci, strepto meaning arranged in a line. Cocci is round, right? We can have tetrad is 4, sarcinae is 8. A staphylococci, it's cocci because it's round. Staphylo is a bunch of them. See how they're all like bundled up here. Bacillus, here's a single bacillus. 2 of them is diplobacilli. A line of them, streptobacilli. Then we have this coccobacillus. So this is actually several, and they are rod shaped, but because they are like squished together, they're arranged in that way, they don't look quite as rod as they would be. For the spiral shapes, we have vibrio. It's just a curve. It's like one that's curved. Spirillum, it just looks like it just has like 1 or 2 twists in it, and spirochete has lots. So those are cell shapes and arrangement. Bacterial species can be either pleomorphic or have many shapes, or monomorphic, which they all look the same. So here H. pylori is pleomorphic. So we see they're not quite as uniformly shaped. Like this one looks kind of squished and round. Here's one that's more stretched out and long. Here's kind of a curved one. So they're not all exactly the same looking, whereas E. coli, they're all look like exactly the same rods. Same size, same shape, everything. All right, let's look at some of these external structures. So these are going to be structures that are outside of the cell wall, and they include the glycocalyx, flagellum, fimbriae, and pili. Let's go through those. Starting with the glycocalyx. So this is a secreted external surface layer that's composed of either sticky polysaccharides or polypeptides. And there's 2 main types. We have capsules, and capsules are more organized and firmly attached to the cell wall. We kind of talked about capsules earlier, how those are going to be surrounding the cell outside of the cell wall. The other type is slime layer. The slime layer is less organized and more loosely attached. But either way, both of these are main function is in attachment. So they can either be attaching them to other cells, kind of bunching them up together. They can also help to attach the cells to the host, or also to like a fomite. This is really important because this is part of biofilms, and biofilms are really important in hospitals because it can be very difficult to sanitize or clean biofilm up. So they're kind of resistant to disinfectants. The glycocalyx also helps these bacterial cells to evade the immune system. So they can, by doing this, prevent phagocytosis of themselves, which phagocytosis is the engulfing of the bacteria by immune cells. So if they evade the immune system, they're not going to be phagocytosed. This is what we call a virulence factor. It makes them more like potent. A couple of examples of species that have these. Streptococcus pneumoniae and Streptococcus mutans. Flagellum, which is the singular word, or flagella, which is plural. These are appendages that are external to the cell, and if present, they're used for motility or mobility. This is another virulence factor. So if a bacterial species has a flagella, it's going to be what we call more virulent. It's more likely to overcome the immune system because it can move around and be more modal to reach areas that's more desirable for the cell. So there's several different types of flagella. So if a bacterial cell does not have any flagella, it's going to be atrichous. A means without, so atrichous is without flagella. Monotrichous, mono means one, so if it just has one flagella, it's monotrichous. A lophotrichous has a lot of flagella coming from one location. I remember that because it's like there's lots of them that are localized in one spot. That's how I remember that. I'll start with L. Amphitrichous has one on each end. Amphi usually means both, so it has flagella on both ends. And lastly, peritrichous is where we have lots of flagella on the entire surface of the cell. There's three parts of the flagella. So the outermost region, which makes up the majority of the flagella, is called the filament. Then we have the hook, it's kind of in the middle, and the basal body down here is what anchors the flagella into the cell membrane. Flagella, of course, are allowing the bacteria to move towards or away from a stimulus. And the way that they do that is they have two different phases. They have a run phase and a tumble phase. And they're either going to run or tumble based on the directionality of their spinning their flagella. So if they rotate the flagella in a counterclockwise direction, that's going to cause the flagella, or it's going to cause the cell to run. So it's going to move in one direction. And then if it switches that rotation to a clockwise manner, that's going to cause it to tumble. And when they tumble, they change direction. It's kind of like a game of red light, green light. So they're going to start running in one direction. Not red light, green light, I'm sorry. Like hot and cold. You know how you play the game, oh yeah, you're hotter, to where it is that you're trying to find. So they start running in one direction, and then they'll stop, tumble, so they're going to change direction, and then they'll start running in that direction. And if they're getting hotter or moving in the direction that they want to be moving in, then they'll keep going, and if not, they'll stop, tumble again, move into a different direction, and start running in that direction, and do it all over again. So if they're getting hotter, they'll keep going, and if they're getting colder, then they'll they'll tumble again. So that's what's happening here. The desired direction is in this general direction. So they ran, and then they tumbled and changed direction, but that was not the right direction. So then they tumbled again, started moving this way, and then they tumbled. And so they keep alternating between running and tumbling. If they don't have an attractant or a repellent, so they don't really have anywhere to go, they just kind of move around in different directions, still alternating between running and tumbling. Axial filaments are still filaments. They're still used for motility, but these are the filaments that are found in spirochetes. So spirochetes are, remember, they have like lots of twists in them. They're kind of long. So this axial filament is going to cause the cell, as it moves, to kind of move in a corkscrew manner. Lastly, we have fimbriae and pili. So these are both kind of hair-like looking. Fimbriae are hair-like appendages that allow attachment. So fimbriae are for attachment. Pili are used mostly for motility. They're not as effective as a flagella, but they can kind of help the cell to kind of glide in one direction or kind of twitch and move a little bit. And there's a specialized pili called the sex pilus, which can transfer DNA from one cell to another. Let's take a quick break here. We'll pick back up when we talk about internal cell structures. See you guys in a little bit.